Good afternoon, my dear students. Here I am once again to deal the chapter the locomotion and the movement. Student, in this chapter I have already completed the introductory part of this chapter. I have completed the types of movement that takes place inside our body. At the same time, I have completed the types of muscles. You have studied about the striated muscle, non-striated muscle, as well as the cardiac muscle. Clear? In my last video, student, I have explained you about the structure of the striated muscle as well as the structure of the sarcomere. Clear, student? In this video, I am going to explain you about the mechanism of muscle contraction. Clear? Student, there is an important theory, that is the sliding filament theory of muscle contraction. हम लोग यही theory पढ़ेंगे. We will study this theory only. Clear? And this theory was given by the A.F. Huxley and R. Niederjarke. Clear? In the year 1954 in England. Clear, student? So before going through this sliding filament theory, I would like to remind you the structure of that actin and the myosin filament. Actin mein, student, you already know that it has two helically coiled F actin. आप देख सकते हो, you can easily see these two are the F actin. Clear? Then F actin is associated with the tropomyosin. Clear? Tropomyosin का structure is there, and troponin is also associated with the troponin. Clear? At regular interval, another protein, that is the troponin, it is also attached. Clear? So this entire structure make the actin filament. Clear? You must be remembering the structure of the sarcomere. Clear, student? Sarcomere mein you have the idea that these two are the Z line. Clear? And these are the actin filaments. Clear? So actin filament is made up of what? Two F actin, then tropomyosin and troponin. Clear? Now about the myosin. Myosin mein you have studied. Myosin mein you can see easily there are two myromyosin. They are arranged helically. Clear? It has the head with a short arm here, and this is the tail. Clear? The head has binding site, that is ATP binding site, and second is the actin binding site. This structure I have already explained you in my previous video. Clear, student? So let me explain you the sliding filament theory now. Clear? So sliding filament theory of muscle contraction. Clear? It is due to the sliding of thin filament over the thick filament. Clear? Here you can see this is the thick filament. These are the thick filament. Clear? And this is the thin filament. Thin filament, or you can say isotropic band. Clear? And this is the thick filament. So what happens during the muscle contraction? These two Z line come closer. Yani you can see these thin filaments slide over the thick filament. Clear? In detail I will explain you. Clear? This happens during the muscle contraction. Clear? And these two Z line they come closer. This is the muscle contraction. So sliding filament theory, it states that contraction of the muscle fiber is due to the sliding of thin filament, that is the actin, over the thick filament. Clear? Up now, what is the first step? What is the first step of muscle contraction? तो first step is the muscular contraction is initiated by the neural signal, that is the CNS. The signal travels, clear, it is generated from the CNS, clear, and through the motor neuron it reaches to the neuromuscular junction, clear. Once again I am telling you, the first step, that is the a signal from the CNS travels through the motor neuron and reaches to the neuromuscular junction, clear student. You can see here, this is the nerve ending, clear, and you can say this is the muscle, this place, this is the neuromuscular junction. Here you can easily see the nerve ending, clear, nerve ending, it secretes neurotransmitters. You have idea, clear student, neurotransmitters are there, clear. So now these neurotransmitters sends the signal here, clear. So as soon as the muscles get the signal from the neuron, once again listen, as soon as the muscles get the signal from the you can say nerve ending, clear, or the neuron, then calcium ion is released, clear. So many calcium ions are there, they are released, clear. कहाँ होते हैं? Where these are? These are there in the sarcoplasmic reticulum, clear. You must be remembering that sarcoplasm which are present in the cell or the fiber, that is the storehouse of calcium, clear. So what is the role of calcium, clear? What is the role of calcium? Let us discuss. First of all, you should know the calcium ion binds. Calcium ion binds. I am just telling you. So this is the troponin, clear, troponin protein. Now calcium ion, they bind to the troponin, clear, and some configurational changes takes place, clear. Some configurational changes takes place in this entire actin filament, clear student. And then what is the result of these configurational changes? What happens? The site which is masked by the tropomyosin, clear, you can say the site of the actin filament which is masked by the tropomyosin, it is now unmasked, clear. Once again you listen, the calcium ion which are released from the sarcoplasmic reticulum, they gets attached to the troponin protein, and this results in the configurational changes that takes place in the actin filament. वो uncoiling हो जाता है, and then what happens, the area that is masked, it become unmasked, वो हट जाता है, it become uncovered, clear. So this happens because of what? This happens because of activation of the, you can say, calcium ion, clear. Calcium ions are released, they get attached to the troponin, clear, and the troponin help in unmasking the actin binding site, clear student. Now what happens? Second step में what happens? Now this head has the actin binding site, clear. Now this head of the myosin binds to the active site of the, you can say, actin, clear. Here you can see, clear, here it will bind, clear, it will bind here, the head, this is the head, it binds to the actin filament, clear. The head of the myosin binds to the actin filament, and for binding what happens? One ATP, it gets attached to the ATP binding site, clear. ATP binding site से ATP attach होता है, then ATP is broken down into ADP and inorganic phosphate. Now the energy which is liberated, it helps in binding the head of the myosin with the actin, clear, and thus a cross bridge is formed, clear, a cross bridge is formed, you can see here, clear student. And now what happens, now stroke, a very strong stroke takes place, clear. Yani first of all you can see the attachment of the head to the actin filament. The head of the myosin gets attached to the actin filament. This is the first step. And here which molecule play a very important role? ATP plays a very important role, clear student. ATP plays a very important role. It is broken down to form ADP and inorganic phosphate. The energy which is liberated, it is utilized in formation of cross bridge, clear student. Now what happens, power stroke is generated. Now it will pull, the head will pull the actin like this, clear. Now this is the, you can say, actin, and this is the myosin filament. The head gets attached. Now it will pull it here, this side, like this. So what similar case occur here also, clear. So now these two will be pulled this side, clear. So this is the contraction, clear student. And this condition persists till the work is done, clear student. Irregular contraction and relaxation, it keeps on going, clear. Yani once again you listen, the myosin head binds to the active site on the actin to form a cross bridge. This utilizes energy from the hydrolysis of the ATP. Then what happens, this pulls the actin filament towards the center of the A band, clear. Now these two will be pulled, I band or actin will be pulled towards the center of the A band. Then what happens, as a result Z lines come closer, these two Z lines they come closer, clear. This results in shortening of the sarcomere or contraction of the muscle, clear. Then what happens, a very important point I am just going to tell you student. During contraction, length of the A band remain unchanged. You can say the length of this band, it remains unchanged, clear. Whereas these actin become short, clear. This is the very important statement that is often asked in the examination, during NEET examination as well as during the board examination. This is often asked, clear student. So now what happens, next step is the, you can say, a detachment. Detachment means what happens, again one ATP is required, to break this this attachment, clear. Again ATP is attached and this is broken down to ADP and inorganic phosphate. The energy which is released, it is utilized to break this bridge. So two molecules of ATP are required, one for formation of the bridge, one for attachment, and another ATP for detachment, clear student. So this was the simple mechanism of muscle contraction. Attachment, clear. Then power stroke, this is the power stroke. This diagram is showing the power stroke. Because of the power stroke, these two, yani thin filament or the I band or actin, they are pulled towards the center of the A band, clear. The length, you can see the length of the A band remain unchanged. Myosin की length ऐसी रहती है, but these two become short and the Z line comes closer. Clear? Through this diagram I am showing you. You can easily see initially these two are the Z line. Clear? This was the initial stage. Now next step में what happens? You can see yani here you can see the distance become very less and these two come closer, Z line come closer, and ultimately what happens? It overlaps. Actin filament overlaps the myosin, and this results in shortening of the sarcomere. Clear, student? So this is repeated many a times. Clear? So it was the simple mechanism of muscle contraction. Clear? Yani a new ATP binds and the cross bridge is broken. Clear? After attachment, detachment is also necessary. Clear, student? So a new ATP binds. Clear? And it is broken down to form ADP and inorganic phosphate. The energy which is released, it is utilized in breaking the bridge. Clear? So this cycle of cross bridge formation and cross bridge breakdown continues till the calcium ions are pumped back to the sarcoplasmic reticulum. Clear, student? So when sarcoplasmic reticulum gets the calcium back, then what happens? Again some changes takes place in the actin filament and it reaches to its original condition. Yani what happens? The site, the myosin binding site, the place where myosin head binds to the actin, it is again masked by the what? You can say tropomyosin. Yani the condition, it is repeated. Clear? Yani original scene, it comes back. Clear, student? So here you can say that the calcium ion play a very important role. So three main steps takes place at the time of muscle contraction. Clear? First of all, attachment, clear, of the myosin head, then power stroke, clear, and then detachment. Clear, student? So I hope that you are now able to understand the sliding filament theory. Clear? Now I would like to explain you about the two types of muscles which are found in the human body. The two types of muscles, they are called as the red muscle and white muscles. Clear, student? Red muscle and white muscle. Red and white. Red muscle fiber and white muscle fiber. Yani what are the differences? Red muscle, it contains more myosin, and white contain less myosin. Clear? This is red in color, this is white in color. Clear, student? Now it has more mitochondria. It has more mitochondria, whereas it has less mitochondria. Clear, student? So these are the basic differences. Clear? Or here you can say it has less sarcoplasmic reticulum, it has more sarcoplasmic reticulum. Clear, student? And since it has more myosin, clear, it can work for the longer period. Clear? I am writing here, it can work for longer period. Clear? And it does not fatigue easily, whereas it works lesser period. Clear? and fatigued very easily. Fatigued very easily. Clear, student? So in this way we have completed the first part of this chapter, that is the structure of muscles. Clear, student? We also completed the sliding filament theory. Clear? In my coming videos, student, I will explain you about the second part of this chapter, that is the skeletal system. We will discuss about the various kind of bones which are present in our body. Clear, student? Till then, you watch this animated video. Clear? Enjoy this animated video. Clear, student? And I hope that you like this session. Clear? So keep on watching this video, student. And next video may I will come with another topic. Thank you. Have a nice day, students.
You use muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.